So with SLAMseq, we have developed a technology that basically combines metabolic RNA labeling with high throughput sequencing to assemble what we can refer to a molecular movie of gene expression as it occurs inside living cells. Now, this enables us to follow the fate of RNA molecules in their native environment from their production by transcription to their sequential processing and maturation up to their tightly controlled decay and turnover. Now with SLAMseq we can precisely quantify these intracellular rates of key steps in gene expression to learn about the fundamental principles of how genetic information is put into practice in a highly controlled manner during cellular differentiation, the response to external stimuli, or the development of diseases. Now, more recently, we have combined SLAMseq with small RNA sequencing to determine the timescales, molecular features, and regulation of microRNAs to understand how these small non-coding RNAs can act as key regulators of organismal development, physiology, and disease. Well, so I'm deeply honored to have received already the third ERC grant, which I think is first and foremost a recognition of the excellent mentorship and education that I enjoyed in the past. But also the outstanding team of scientists that I was lucky to recruit over the past years into my lab. And I think it is likewise also a recognition of the excellent research and support infrastructure that is available to us here at IMBA. Now, I believe that at the core of this success is also the extraordinary collaborative spirit at the Vienna Biocenter, where highly diverse research areas are brought together in 4 basic research institutes. And I think with meanwhile more than 50 ERC grants that were awarded to scientists at the Vienna Biocenter, it ranks among the leading biomedical research centers in Europe. And finally, I think that this recognition is also a testimonial for the overall impact of our research interests that centers on the versatile function of RNA molecules that have already puzzled and surprised many generations of scientists. And in future, I hope that we can continue to contribute to the established status of Vienna as a hotspot for RNA research in Europe and beyond. Mm-hmm. Now, with support of the ERC Consolidated Grant, we will investigate the least understood aspects of gene expression, namely the regulation of RNA fate and function. Now, our goal is to understand the molecular mechanisms that control the expression of genetic information, which is the fundamental process that unites basically all living organisms. And as such, our work will most certainly impact a broad array of biological processes and will have direct implications for our understanding of human diseases and the development of therapeutic approaches for its treatment.